Hello guys, my name is Shankar S56434 and today I will be presenting about understanding of diversity marine phototrophs. For the introduction, traditionally we believe that the diatom and flagellate are the only marine primary productive, but as the technology became improved, we realized that there are many more marine primary productive other than the diatom and flagellate such as phototrophs. So, in 1970s, we believe that diatom and flagellate are the only marine primary productive. This is because they are widely distributed. We can find them in the eutrophic and oligotrophic environments. Secondly, because of the sampling and method of phytoplankton detection. Traditionally, we use the phytoplankton net with the size of bigger than the 60 micrometer. So, this makes that phytoplankton that can be cached are only in that range. So, as we can see on the picture on the right, there are phytoplanktons that are smaller than the 60 micrometer. For the current understanding, the usage of the DNA detection and the flow cytometry as shows us that there are phytoplanktons, there are planktons that are smaller than the size of 60 micrometers. Okay. For the recognition of the phycocyanobacterial dominance, in the late of 1970s, the observation technique which I use that is fluorescent microscopy and electron microscopy observation as shows us the unicellular cyanobacteria named as Cyanococcus. So the characteristics of this Cyanococcus is it is 0.5 to 2.0 micrometer in length, 0.5 to 1 micrometer diameter. So it is classified as a phycocyanobacteria. So in the late 1980s, the usage of high performance liquid chromatography HPLC and cell detection technique using flow cytometry has brought us to the new Phycocyanomacea genus, which is Prochlorococcus. So what differentiate with the Prochlorococcus and Cyanococcus? So the Prochlorococcus contain the divergent chlorophyll DVCHL and do not contain picosbilipigments, which is a light harvesting antenna that is commonly found in the Cyanococcus. So, it Pro Prochlorococcus is a coccoid to the ellipsoidal unicellular cell. It performs a oxygenic photosynthesis, which means that the byproduct of the photosynthesis is the oxygens, using the divergent chlorophyll A and B, which is efficient in absorbing the blue light. So, the discovery of ubiquitous photoheterotrophs. Photoheterotrophs are the organisms which can use light energy as their energy for their energy source, but they cannot use carbon dioxide as their sole carbon source. So, there are two significant findings in the early of 2000s, which is first is the AAPB, which is the aerobic anosogenic photosynthesis bacteria, which contain bacterial chlorophyll A, and the second is the distribution of the proterodopsin containing bacteria. So, what are those? AAPB, which is used bacterial chlorophyll bichlorophyll A for their photosynthesis, as they require oxygens and they fulfill their carbon requirement by absorbing the dissolved organic matter in their surrounding. So for proterodopsin, it contains a rhodopsin-like protein which helps them in detect the light and transforming the light into the ATPs. So oxygenic photosynthesis using far red light. The in situ light absorption range by the chlorophyll A, which is from 400 to 700 nanometer, is known as a photosynthesis active radiation (PAR), which is used to evaluate the light strength for the activity of the oxygenic photosynthesis. So in 1996, it is discovered a cyanobacterium named as Archaeochlorus marina, which can perform a oxygenic photosynthesis using a chlorophyll D. So this chlorophyll D has a unique characteristic where It can perform oxygenic photosynthesis using a far-red light which has a spectrum from 700 to 740 nanometer. So, in 2010, a red-shifted chlorophyll F was discovered in the coast of Australia. It has the observation spectrum around 720 to 760 nanometer. So, this is a research done by the Savishi and Chen in 2020, where it shows that the chlorophyll D and chlorophyll F uh, help the cyanobacteria species to grow in the shaded environment and establish a unique habitat. In 2010, uh, several several reports has been done by using the technique of metagenome and flow cytometry, where it discovered a significant amount of phycohaptophytes, including many as described species. One of it is the phycoplankton fraction, which have a carotenoid 9 pigments which only can be found in the haptophyte. So it shows that 50% of the total amount phycophyte is belong to the haptophytes. In 2007, the metagenomic analysis is once again found a new phytophyta which do not belong to any phylum, named as phycobiliphytes. In 2011, a new candidate phylum was also detected by the same technique, which is a metagenomic analysis, which is named as a Rapimonads. It is close to the Aptophyte but did not belong to any known phylum. So, the cyanobacterium-like particle UCYNA was also detected by the metagenomic analysis. Usually, cyanobacteria grows by an oxygenic photosynthesis. Sorry, sorry. Usually, cyanobacteria grow by an oxygenic photosynthesis, but this UCYNA is lack of photosynthesis to rubisco and the primary enzyme for fixing carbon dioxide and the terricarboxyl acid. So this lacking of these essential genes for living suggests that the UCYNA might be a symbiont in some organisms. So, the current understanding on the diversity of light energy transformation system and reconsideration of photosynthesis suggests that has shown that the phototrophs are the organisms that can grow using light as its source of energy, while photosynthesis is the process of reducing the carbon dioxide in the environment by transforming it into organic compound. And the primary producer are the organisms that can synthesize organic compound using inorganic carbon as its carbon source, and it can happen without the presence of light. For example, chemoautotrophs. So this is the timeline on understanding of diversity. First start with the traditional, which is start at 1970, which is a uh, which is the understood of the diatom and flagellate as the main primary production, and at the late of 1970s is the discovery of phycocyanobacteria. At the 1979 is the discovery of APB, which is a anosogenic photosynthesis bacteria. In 1980 is the discovery of phycocyanobacteria, which was named as Cyanococcus and uh, Prochlorococcus. In 1996 is the discovery of Cyanobacteria Acrocloris marina, which have chlorophyll D. In the early 2000 is the finding of the two significant photoheterotroph, which is the AACP and the Proceroadopsin. So in 2007 is the finding of the phytoplankton which do not belong to any phylum, named as the phycobiliphytes, and in 2010 is the report on the phycoplankton using the technique of metagenomes, and the discovery of the red-shifted chlorophyll F, and that in 2011 is the new candidate phylum which was detected by the metagenomic analysis named as the Rapimonas. So that's all for me. Thank you.